Hi. So it's a great pleasure that I can introduce Georg Oeltzschner. So I practiced that last name for like 2 weeks. So he received his Ph.D. in physics from the University Düsseldorf in Germany. His supervisor was Alfons Schnitzler. And he's currently a postdoc at Johns Hopkins University School of Medicine in the lab of Richard Eden, and he also works very closely with Dr. Peter Barker. So his research is dedicated to advance the MR spectroscopy method for the detection of low concentration metabolites such as glutathione or GABA. And his talk is called New Frontiers of Edited Magnetic Resonance Spectroscopy.
Well, thank you for having me here. Thank you for coming. And just as a preface, if I say anything that's completely not understandable, just please raise your arm, raise your hand, shout, do anything. I'll try. Like what I want for everyone to like, get out of this talk is get out of this room and have learned a tiny little bit. And even if it's just a very basic thing, and if well, I struggle to explain that precisely or nicely, then please stop me in my tracks and feel free to ask any question at any point during the talk. Don't hold up until the end, and just go with it. I am on the train here. I did a little bit of research about Eva's background, and I found out that Eva and I actually, well, not from not very far apart in Germany. This is pretty much where I grew up. It's a very rural area in Germany. After that, after growing up in that very rural area, I made it to Düsseldorf, into the big city. I was surprised there were trams and everything going, public transportation. That was great. Finished my PhD there, and for the last 4 years, I've been here at Johns Hopkins Hospital in beautiful Baltimore. And I'm finally back in Boston after 3 years now, and I'm super excited about it. I don't think I've ever made it here when it wasn't below zero, no, below freezing, below 0 Celsius. So cool to be here, and I'm excited, and I'm hoping that I can teach you a little bit about the exciting technique that is MR spectroscopy, what we can do with it. The talk is briefly, broadly divided in 3 talks. At first, I'm going to talk about what is edited MR spectroscopy. So a lot of you apparently have a little bit of, heard of MRS, what it is and what it does. We're going to talk about what we are specifically doing in edited MRS. Going to look at a couple of example applications. What is this useful for? What can we learn from it? What can't we learn from it? Finally, quite a large part of this whole thing is going to be dedicated to a couple of things that we've worked on recently, trying to make MRS more exciting, more versatile, and the most recent efforts also more standardized and more public. And we'll hope that you may enjoy it. Just as a note ahead, almost everything that I'm going to talk about is going to happen at 3 Tesla. I know that you have a bunch of 70s standing around here, but 3 Tesla is still the most widely used one, obviously. So we're talking about this. So obviously many of us use MRI on a daily basis, right? Some MRS, so it's great, you have a head start. And in proton imaging, so almost everything that we do is based on measuring the signal that we receive from the water protons, right? So they resonate at a certain frequency. If we plot the signal from the brain over the Larmor frequency, we'll see something like this, right? But water is obviously not the only compound in the brain that contains protons. So if we zoom into this seemingly empty area of the spectrum and we suppress the water prior to our acquisition, we will see something like this, right? So this is a typical MR spectrum that we could see recorded from a brain, and it contains signals from metabolites that also contain protons, but at a concentration of like 5 orders of magnitude lower than that of water, which is in the millimolar range and which complicates matters a lot, as we will see. So first of all, why do these protons have different frequencies? And the answer is that each group within a molecule has a characteristic chemical environment, right? So the electron densities slightly modify the magnetic field at the spatial location of the proton, and that gives rise to slightly different frequencies that we can record here, right? And we can immediately see that we can directly quantify, if that laser pointer works, yes, some of the large signals. So there's, for example, N-acetyl aspartate here, there's creatine here in red. But everything else is very complicated to assess. It's all down here in the weeds. There's a lot of overlap. And just as an example, one really interesting compound that neuroscientists and clinicians like to look at in the brain is GABA, which is gamma-aminobutyric acid. It's an inhibitory neurotransmitter. And see whether you can spot the signal from GABA. So it's obviously somewhere down here. Let's take a look where it actually is. So we take all the other metabolites away. And GABA has concentrations between 1 and 2 millimoles. So it's these small green humps here at 1.9, 2.2, and 3 ppm, and each resonance is buried under huge other signals, right? And for reasons that will become clear in a second, the signal that we're most interested in is at a frequency of 3 ppm, so it's this one here, right? And the signal overlaying this resonance is this massive creatine hump here. And at standard clinical field strength, it is impossible for us to reliably separate GABA from under there. So the big question is there a way that we can, is there any way we can get a hold on this tiny green signal down there? And the answer is, fortunately, yes. Otherwise, I probably wouldn't be here. But in order to understand how this works, we'll need to have a closer look at the GABA spectrum first. So as we see, GABA has 3 proton groups here. This one isn't depicted here. Each resonating at different frequencies, and this is what it looks like at an echo time of 80 milliseconds, right? So you see there are signals from 3 proton groups within the molecule. The pink protons here produce a signal at 1.9 ppm, and the red protons here produce a signal at 3 ppm. Let's for now not consider the 2.25 ppm signals. And since the pink 1.9 ppm protons are located very closely to the red 3 ppm protons in space, they inflict a small local change in the magnetic field at the location of the red protons and vice versa. So the protons feel each other, you could almost say. And this is a phenomenon we call scalar coupling or J-coupling. And we will see in a moment how we can utilize this intrinsic molecular property to our advantage. Again, this is the GABA spectrum from the previous slide. If it would look like if we measure it with an MRS sequence, and this orange area here highlights the signal that we want to look at, right? It's the 3 ppm signal from the red protons. And now, during the acquisition of this spectrum, we can externally manipulate the configuration of the molecule, the electronic configuration of the spin system, with a selective radiofrequency pulse. And this pulse is applied at a frequency of the 1.9 ppm protons, right over there. And what it does, it flips the orientation of these protons relative to the external magnetic field. But thanks to the coupling, because these protons feel each other, this minimal change in the configuration of the protons acts on the 3 ppm protons. And what happens is when we do that, is that the 3 ppm signal changes its shape. So the outer wings of this signal, they flip upwards. And this is why we call this an editing radiofrequency pulse, right? Now, what does this mean for us? The use of this will be immediately clear as soon as we take a look at the MR sequence with which we acquire our signal, right? So the way that MRS acquires signals, generally, first of all, we localize the origin of our signal, well, in the slice that you just saw here with a slice selective 90-degree pulse. Then we add another slice selective refocusing pulse a bit later with an orthogonal slice selection gradient to that to select a slab. And after half the echo time, we add another slice selective refocusing pulse in the remaining slice selection direction, and that reduces our excited volume down to a cube, right? So this is a typical volume that we acquire from to do these GABA signals, like a 3 by 3 by 3 centimeter cube, in this case from posterior cingulate cortex, and this is pretty standard for us. You can go smaller. I'll talk about that in a little bit. So if we do exactly this with this sequence, we just acquire a normal spectrum, but we also run the exact same experiment again, but this time we apply these editing pulses to change our signal. Has anyone done here ASL, or is anybody familiar with what ASL? So the underlying idea of doing 2 halves of an experiment, if you want to detect something that's really tiny and is buried under something huge, the general idea is you do a tiny little bit of a change to the thing that you want to detect, and then you can subtract out the background that isn't affected by this manipulation. And we're doing this exact same thing. So we're doing a normal spectrum and the exact same experiment, only this time with the editing pulses. So we have 1 spectrum with the editing pulse on and 1 spectrum with the editing pulse off. And this is the so-called, the famous mega press sequence, or the mega editing sequence. So we have 2 halves of the experiments now. What do we do with them? And the answer is obviously we subtract them, right? And we do that because the key to isolating this GABA signal in our in vivo spectrum is the fact that the editing pulse specifically targets the GABA molecule, but nothing else. If a molecule does not contain a resonance at 1.9 ppm, the signal shape will not change. The editing pulse will go right through it, and the molecule doesn't even feel that the editing pulse is there, all right? So if we perform 1 acquisition with the editing pulse on, we flip the signal here, and we perform 1, oops, that goes too fast. We perform one without the editing pulse applied here, and we subtract these two spectra, then only the compounds that are affected by the editing pulse will contribute to the different spectrum right here. Right? So we see that the green GABA signal is retained, but the red creatine signal is the same in both acquisitions. It's not affected by the editing pulse at all, so perfectly subtracts out. Right? It's the same as in ASL, where you label your blood once and you label your blood in the other experiment you don't, and you subtract the entire background out, and you only get a blood image. Right? We call this different spectrum the GABA-edited spectrum. And in our case, we are most interested in this isolated 3 ppm signal. And let's have a closer look at it, what it looks like. So this is, if we zoom into it, it has contributions from all the molecules that were affected by the editing pulse. And that could be through the coupling, like the GABA. It can be directly, like the N-acetylase signal, or indirectly, like the glutamate, glutamine, and glutathione signals, which are chemically quite similar to GABA. Right, as I said, we're mostly interested in this little hump here. And the area under the peak is directly proportional to the concentration of GABA within the cube that we just excited. So all the molecules within this volume. We'll get back to this in a little bit. This technique is super sensitive to a lot of hardware and subject instability. So some post-processing is always needed in order to optimize the data and to quantify it accurately. We need to experiment, or we need to repeat these two halves of the experiment about approximately 150 times in order to gain enough signal to noise, in order to have our signal rise out of the noise. So it's very repetitive. And there is some post-alignment and corrections to do in order to retain the optimal signal. We have a free MATLAB toolbox, it's called GANET, that we've specifically developed in order to quantify these signals, right? It's a relatively trivial signal model here. We just fit a Gaussian through this different signal that we saw. And it quantifies it relative to an internal standard, right? That is believed to be stable or has known concentrations, such as the brain water or such as creatine, and sometimes even NAA. Let's briefly talk about how much time we need to acquire a different spectrum like the one that we just saw, because this is a major limitation of the whole technology and the technique, and that we're trying to address with our modern methods. As I said, we're looking at signals with much lower concentration than water. And so a single shot acquisition almost disappears in the noise, so we have to average a lot. So these are typical parameters for the acquisition. We have to average over 300 times, resulting in roughly 10-minute scans. We're looking at relatively large volumes, and the question that we always run into from collaborators is, can we make our voxel smaller? We just had this discussion earlier with Marco, who is an applications person. He wants to make his voxel as precise as possible and as small as possible, and be, well, you can do that, but you're not going to be very happy if you make it too small, because signal to noise is directly proportional to the volume of the voxel. But if you want to buy signal to noise back, it's going to cost you, and it's going to come to bite and to come back to bite you, because signal to noise is only proportional to the square root of the number of averages. So if you want to gain spatial sensitivity, you pay an extremely high price. So let's say if you want to halve your voxel size, you'll halve your SNR, and you need to scan for 4 times as long in order to compensate for the loss in signal to noise ratio. So you can do that, but it's just going to diminish your data quality. All right, that's for the quick recap for the 13-minute run through. How do we, and what do we measure with MRS at this point? Take home message for now. If you don't take anything home, we can measure GABA in the human brain in vivo at 3 Tesla in about a 10-minute scan, but it's not easy. That's the takeaway, I think, from the first couple of minutes. Are there any questions so far until now? Great, so I could have a drink. Yeah, perfect. Wonderful. All right, thank you. It's either a good sign or a really terrible sign if there are no questions. I hope it's not the worst. Right, now that we have some rough idea about what we're supposed to do, we can have a closer look at what we can do with it. So, please. Probably not.
So how accurate?
Define accurate. Define. There's a difference between accuracy and precision.
Correct. Okay.
Yeah.
So just B-test. So what is the variance?
On the order of magnitude of 10%. So it's not terrible. It's also not great. Like, we would love it to be more precise. We don't know a lot about the underlying fluctuations. for example, there are a couple of papers that show a dependency on menstrual cycle. It's never been reproduced because nobody has ever looked at it again. So might be, might not be, might be a source of variation, might not be. It's not well studied. And part of the variation is obviously stems from your modeling. Part of it stems from your acquisition. How well are you able to repeat that exact placement of your measurement volume? You know, there might be some variation associated with that. GABA might change over daytime. There are many reasons why there might be fluctuation that we can't see. Or both, yeah. It also depends on your reference compound. That might also change. Yeah, there's a bunch of it. But overall, coefficient of variation, to answer your question in one sentence, about 10%, and that's been shown to be relatively stable. Sorry, say again. If you make a phantom of a complex, yes. Yes. You can get arbitrarily precise with a phantom. Yeah, like what? Like a 5%? Lower than that. Lower than that. You will. In the problem with the in vivo measurement is partly that we do have some sort of signal contamination from other compounds. So what we're measuring is technically GABA plus a couple of other things. So that's one tiny source of inaccuracy that we get there. The modeling in vivo is a bit harder than the modeling in a phantom. I can analyze phantom data relatively easily if I push them through an algorithm. With in vivo data, there's a lot of background from macromolecules that goes on that also makes puts the accuracy of the whole method a little bit into question. So the question or the distinction between accuracy and precision is very important. I think we can do it relatively, well, we can do it reproducible. The question is really how well do we nail the amount of GABA that is actually in there and what's the meaning of it? But we'll get to specifically the second part of that right now because it's quite an interesting, the maybe more interesting question is how do we interpret what we measure? So we'll get there. Was that a helpful answer, Eva? Was that a helpful answer so far? Sure, yeah. That sounded not very convincing. What I really want to know is, you know, if you can actually partition the errors in that to subject movement, you know, temperature changes of magnet. Well, from a single measurement, we can't. We can assess how susceptible are the quantitative measures that we get to the factors that we just saw. So if I see a measurement and I know that the scanner has been drifting a lot in its frequency, then I can make a well-educated estimate or guess about how much that will affect my measurement error. That is possible, but it's not always easy to tell drift from subject motion or frequency instabilities from subject motion. That is not always possible. You can measure that. Yeah, we do, and we do. That is part of our software. We compensate for that. All right, should we go on? So generally, we have two pillars of specifically GABA MRS application in the human brain so far, and one of them was it's pretty widely used in the healthy brain, just in cognitive neuroscience, sensory processing, perception, behavior, performance, and how all these measures relate to the static baseline GABA levels. And it's obviously also very interesting to look at situations where GABA levels may change. And the first thing that comes to mind is disease. So maybe can help us understand pathophysiological mechanisms by asking how are GABA levels related to clinical symptoms or genetic mutations, for example. How do GABA levels change upon intervention is another interesting question, right? There's a lot of studies that look at pharmacological interventions, TMS, brain stimulation, you name it. And where the circle closes back is how do modified GABA levels through intervention reflect back on behavior and performance. So it's kind of, it connect like a nice bridge between different levels of assessing a certain phenomenon, right? But the first thing, as I said, that we actually need to ask ourselves before we can talk about what we can do with it is what the hell are we actually measuring? So with MRS, we're measuring all of the GABA within the predefined region of interest, all of it. Obviously, if we have a large number of neurons in our voxel, we'll see more synapses, we'll likely see more GABA, right? In fact, we do see gross differences between gray matter and white matter in GABA content. So when we quantify our results, we usually correct for the fractional compositions of gray matter and white matter in our voxel. If we look at identical regions, then GABA reductions with MRS most likely reflect actual production of GABA itself from glutamate. And that happens through GAT65, it's glutamate, carboxy, ugh, I always mix that up, which it's the enzyme that synthesizes GABA from glutamate. It sits predominantly in the terminals in the case of GAT65, GAT67, which is rather ubiquitous and produces the majority of static GABA. So if a lot of GABA is being produced, we will also detect a lot of GABA. That's in the nature of the thing. So for example, GAT67 knockout mice, they will have almost no GABA and they will die relatively quickly. GAT65 knockout mice, they survive, but they have seizures. There is a rare genetic disorder, stiff person disorder, where patients suffer from muscle rigidity and seizures and spasms. And 80% of them have GAD antibodies, right? So the GAD protein, the one that synthesizes GABA, is being killed off and less GABA is being produced. And with MRS, it has been shown that these have substantially lower GABA levels, right? So it seems to be reflective, actually, of a static background baseline GABA concentration. However, the issue can also lie with GABA transport, either back presynaptically into the neuron or into the astrocytes. So these things can be modified as well in disease, and they can be pharmacologically modified. So these can all have an impact on levels of GABA. I think the GABA trainers, for example, the GABA transport blocker, which will cause GABA levels to increase. Finally, GABA might be affected in the GABA receptor system. So there we have the faster acting GABA A receptors and the metabotropic GABA B receptors. It's not to say that these don't in some way affect GABA production, but the relationship there is less clear. Generally investigating GABA function as a punchline, it is difficult and interpretation is required. That said, MRS does allow for investigating whether GABA levels are grossly altered, which does point to specific issues in terms of neurotransmitter availability, I guess, regional specificity. And what we like to think about it is kind of, it's a measure of the GABAergic tone, kind of the availability or the potential to exhibit inhibitory activity. So it's important to be careful in interpreting the GABA signal. It's insensitive to whether the GABA molecules are in the synaptic terminals on the cleft or elsewhere. We're not measuring active GABAergic neurotransmission. We're not measuring inhibitory activity per se. We're not measuring receptor density or anything. We're measuring baseline GABA molecules in this region. And I've seen all of these interpretations in GABA MRS papers. So the bottom line is I think it's a useful tool, but still we need to be a little careful about what we're doing with it. It's been associated with the expression of the GAT1 gene, which encodes for GAT67, which is the GABA-producing enzyme. And as I said, the takeaway message is probably, it is rather reflective of a GABAergic tone, a backline potential to exhibit inhibitory activity, which still has a lot of functional and behavioral impact, as we will see. Any questions about that? Cool. So a nice example for how to apply this is a study that my supervisor did during his postdoc time in Cardiff from a while ago. They wanted to know whether GABA levels in the visual cortex have an impact on a specific visual orientation discrimination task, right? So the task is relatively widely used, but still, as a reminder, you as the subject are being presented two black and white gratings like these here, which are slightly rotated against each other. And by pressing a button, you're supposed to indicate whether you perceive this rotation as clockwise or counterclockwise. And this is done in a staircase design so that we can determine your individual rotation threshold, where you are able to reliably and correctly recognize the direction of this rotation, right? Additionally, gamma spectroscopy was performed in the occipital part of the brain, as we can see here. Indicated by the green boxes, and it was done in 15 subjects, and the result is quite fascinating because the individual GABA levels predict the individual orientation discrimination threshold, and the better results, so if you're able to resolve the degree of rotation better, was associated with higher GABA levels, right? Which is, well, being interpreted as a function of being able to perform a higher degree of lateral inhibition, which helps sharpen the processing of your stimulus, right? We will see that in a couple other tasks as well. So we see that frequencies of stimuli are similarly encoded through GABAergic mechanisms in other sensory domains, such as tactile processing. So this is something that is quite well known. Spatial stimulus discrimination worsens when the GABAergic neurotransmission is disrupted in the animal model, right? And that's because we have less GABAergic neurotransmission, we have less lateral inhibition, and as a consequence, we are less able, and this, I think it's a cat paw, so the cat is less able to reliably separate two very closely neighboring stimuli, right? So what happens if that, what also happens is that encoding of frequency information in neuronal activity gets lost if GABAergic neurotransmission gets disrupted. So it seems GABA is critical to encode frequency as well, and one can hypothesize that GABA levels influence the individual ability to discern different frequencies of tactile stimulation just as much as it was in the previous visual example, where it predicted how well we're able to differentiate differential visual stimulations, right? We can probe this individual discrimination ability with a vibrating stimulator on the human finger. And here again, the subject is asked to provide a response on which of these two closely following vibrational stimuli they receive on the finger had the higher frequency. So again, it's a discrimination task that will rely on the way that we are able to exhibit lateral inhibition. And my colleague Nick Putz, he did this during, I think here, during his postdoc at Hopkins. He performed this task in a number of participants and GABA spectroscopy in the sensory motor area, but also as a control region in the occipital cortex. The finding is quite consistent with our hypothesis because the more GABA that we had in the sensory motor area, the lower the frequency discrimination threshold was. That means there was a better performance in discriminating stimuli that had similar frequencies. So you were able to tell a 5 Hertz from a 9 Hertz stimulation, whereas a person with less GABA wasn't able to do that. Interestingly, there was no correlation with GABA in the visual cortex. So there is a region-specific functional impact of GABA levels. And there doesn't seem to appear a uniform, like, tonic inhibition level that is the same throughout the brain, but rather we're talking about region specificity and task specificity.
Did you also measure glutamate at the same time?
Yes. I'd have to go back to that paper to see what they found. I believe that there was no direct link with glutamate, yeah. Because the mechanism is most critically relying on the lateral inhibition mechanism, which is GABA-mediated.
Yes, I mean, it's kind of weird because only a small fraction of the GABA you're observing is actually.
That is true.
So you would expect the GABA would just reflect the overall tablet. Better with visual discrimination than glutamate.
That is, yeah, I don't disagree. I don't disagree. We can talk about it at the end of it, happy to. Let me give you for now an example how we can run these kinds of experiments also to bridge over into clinical research and maybe help investigate pathophysiological mechanisms. So this is a study that I did during my PhD in Germany, and that was in patients with hepatic encephalopathy, which is a complication of liver cirrhosis. So we had a large cohort of patients with liver cirrhosis. So the liver loses its ability to filter toxic substances, and among them the most prominent is ammonia. It does the most damage in the brain. It's a small molecule, can easily go through the blood-brain barrier, right? And it interacts with almost every system in the brain. Symptoms are cognitive and motor dysfunctions of varying severity. There's the so-called minimal or subclinical HE, where the patients appear normal in everyday life, but show impairments in psychometric testing. And then there is, with increasing grading, HE0 to HE4, which starts with mild memory problems and then just goes on until, well, hepatic coma. There is a quick routine clinical test that is used to getting a first idea of the severity of HE, and the so-called critical flicker frequency test, which is again a frequency discrimination task, right? So it's a bit like the two that we've seen earlier. You put on a mask like this and you see a red dot, and the red dot oscillates at about 60 Hertz. So you usually perceive it as a steady light, but now the frequency decreases slowly until you're seeing something like this. So at some point the light begins to flicker because you can actually resolve the flickering that's going on, and you're instructed to press a button as soon as that happens. So this is obviously a measure of the temporal resolution of your visual system. And if you're healthy, you'll be able to discriminate higher frequencies. So if you're diseased, this flicker frequency will go down. And that's precisely what we see. So this frequency decreases with increasing severity of liver disease, meaning that the temporal resolution of the visual area in patients worsens. And well, given that we've seen that GABA seems to be able to predict the performances in these kinds of tasks relatively well, we wanted to have a closer look at this. And we did exactly the same. We looked at GABA levels in the visual system, and we performed the flicker frequency test. We also sampled the patient's blood in order to determine their ammonia levels. And the results aren't even so surprising anymore. So patients, even the ones that didn't have any overt symptoms, but only some early impairments, showed markedly decreased occipital GABA levels. And as we hypothesized, occipital GABA also predicted the critical flicker frequency, with more GABA, again, predicting better frequency discrimination performance. And again, GABA is also, that is very interesting, GABA is directly correlated with the ammonia load in the blood, indicating that the degree of liver damage has a direct impact on brain GABA levels, which we found quite exciting. It's a nice example for what they call the gut-brain axis. This thing gets in the way a lot. Okay.
Very cool results. How do you
Yes.
explain this happening for the larger variants?
Because the disease is very variant, and the grading isn't perfectly accurate. It's there's a high fluctuation in this disease as well. So patients come in and they might be degree 2 at one day, and they get lactulose treatment to flush out the ammonia out of their system. The symptoms get better immediately within 2 or 3 days, and they change back to minimal AG. It's not always the case that this grading is perfectly accurate here. We also see in healthy people, it also was a large age range, and we know that GABA levels change with age. So these were patients somewhere between 25 and 75. So there's also there's certainly a contribution of the spread of ages that contributes to that. There were no differences in age, no. Yeah, people have used it in pretty much any neurological condition that you can look at, and there's always changes in GABA being seen. ADHD, ASD is a very big area of GABA-edited research. There's a bunch of mouse models, for example, that suggests GABA-edited malfunction in autism, depression, bipolar disorder, ALS, you name it. If you search GABA MRS on PubMed, you will find something that matches your area of expertise pretty well. All right, just a quick recap of that part. We can see that GABA levels are a measure of all the GABA molecules in our voxel. Nevertheless, even though it's a very coarse measure of GABA concentration and not in any way localized or specific, as you correctly pointed out, they still do correlate with a lot of functional and behavioral metrics. So we hope that it can still serve as a tool to understand better what's going on in physiology, but also in pathophysiology. Any question about that so far before we dive a little bit into our most recent technical advancements? Flattened. Amazing. All right, cool. So now that we know that obviously this can be fairly useful at all, I want to spend a couple minutes here showing you some of the exciting newer developments that we've come across over the last couple of years in our lab. They all have in common that we're trying to overcome the greatest disadvantage of this technique in general, long scan times. Right? It takes us forever to get one meaningful spectrum. We don't get a lot of data out of a single acquisition, and we just want to be a little more efficient, right? So, first of all, we've only talked about GABA so far, but there are other quite exciting molecules that we can edit the spectrum for. There is, for example, glutathione, which is the primary antioxidant molecule in the brain. And its levels are believed to be a marker of oxidative stress and neuroinflammation, potentially. Even Marco might elaborate to that a little bit more later. 2 mechanisms that are hypothesized to be involved in a lot of neurodegenerative diseases. There's lactate, which is the anaerobic product of glucose metabolism and maybe altered in for example, Alzheimer's disease, maybe an indicator for that. There's N-acetyl aspartyl glutamate, which serves as a glutamate reserve and is a modulator of glutamatergic neurotransmission. Its roles are a little bit unclear, but it's a huge thing that schizophrenia researchers are looking at right now. Then a bit off the immediate charts for us, but for other reasons super interesting, is 2-hydroxyglutarate, which is something that is produced exclusively in tumors that have a mutation on the IDH enzyme, which is involved in the TCA cycle. And this mutation is associated with higher life expectancy because the tumor is more receptive to therapy. And 2-HG is only produced in these kinds of mutations. So the idea is that it's potentially possible to do some sort of like an MR biopsy to determine the mutation status without actually having to go in every single time and pluck some brain tissue out. There's also aspartate, which is another neurotransmitter, and ascorbate or vitamin C, which is another antioxidant compound. So all of these things can be looked at with slightly adjusted edited spectroscopy. And the problem that we're always facing, as I said, is pretty much twofold, right? First of all, itself, MRS just takes a freaking long time. We need to take a lot of averages to obtain enough signal. And that is all the more true for the small coupled signals that we're trying to focus on. There's a little bit of progress in sequence design, hardware design, but we're not going to change physics fundamentally. So we're always going to like pick about the needle in the haystack. The second thing, editing nowadays, up until a couple of years ago, is still pretty much done as it was 25 years ago, or more than 25 years now. And the first paper was demonstrated by the Yale group to measure GABA in vivo in the brain. And this experiment had 250 averages taken with like a 3.5 seconds TR. So that was, again, over 10 minutes roughly. So we haven't made much progress on that side. Generally, the question that we're trying to answer, if we have to scan for 10 minutes because we can't beat physics, can we do more than one useful thing at the same time? So can we throw away as little information as possible so that we're getting as much information out of it as we can? And the first question that we asked was, can we measure GABA in 2 locations at the same time? And for this, we leaned on the so-called PRIAM method that had previously been published at 70. And what we do is pretty simple. We just use a dual band excitation pulse to generate signal not only in one region, but in 2 regions at the same time. And what we do then is pretty much known from imaging. We make use of the parallel imaging technique called SENSE. So it's, we're using the spatial coil sensitivity maps to essentially tear our signal apart and tell where it exactly comes from, because we know that the signal that is generated in this voxel is going to be stronger in this receiver coil. For example, the signal that's generated here is going to be stronger over there. So we're able to disentangle the signals and correctly assign them to their voxels of origin. And we've done this in humans. We've performed GABA editing with this multi-voxel technique in the left and right hemisphere. And as we can see, we get nice GABA-edited spectra at full signal-to-noise ratio. So we get 2 of these spectra in the same time that it would normally take to acquire one of them, which already amounts to a 2-fold increase in data acquisition rate. Which was the first part of our endeavor. How do we make our editing more efficient? That was a spatial domain of this undertaking. But we also tried to get information from more than one metabolite at the same time, right? We've just seen that we can edit for more than one compound. Can we do this simultaneously, or do we have to do one tailored experiment for each metabolite that we're interested? So let's quickly look back. This is the megapress experiment to detect GABA. These are the first 4 averages, right? So we do 1 with the editing pulse on, 1 with the editing pulse off, 1 with the editing pulse on, right? We do this 300 times, and at the end, we will just do the difference, and we'll get this GABA-edited spectrum. We can do the same thing to detect glutathione, but glutathione has a different chemical structure, and in order to detect that signal, we just apply the editing pulse at a different frequency. So it's at 4.56. We do this, we don't do this, we do this, we don't do this. In the end, we take the difference, and we detect the signal at 3 ppm too. Now, the good thing is that these 2 experiments happen at opposite ends of the spectrum, and they're completely independent from each other. So instead of doing on-off, on-off, on-off for each of these, we can squeeze all of that into a single 4-step experiment. I'm not going to go into too much detail because the reconstruction of it is a little bit more tricky. But in the end, what we arrive at is we get GABA-edited signals and GSH-edited signals in one single acquisition at the same full signal-to-noise ratio. It's as if we are performing two of these experiments at the same time, because the GABA molecule doesn't feel what's happening to the glutathione molecule, and the glutathione molecule doesn't feel what's happening to the GABA molecule. So these experiments are completely independent from each other. And that is, again, a twofold increase in data acquisition rate, since we take only one 11-minute experiment, where we would have previously needed two of them sequentially. We don't have to stop there, because we can combine these two techniques. So we can gain the spatial acceleration from acquiring it into regions at the same time, and we can gain the metabolite acceleration from doing GABA and glutathione at the same time. So we get 4 spectra in the same time that it would take normally to get a single one of them, which is a 4-fold acceleration, obviously, in data acquisition rate, which is already pretty cool. So we've managed to up our data acquisition rate by a factor of 4, which is useful. But recently we've taken it a step further. And first of all, we took a step back because we compiled a list of all the metabolites that have so far been looked at with spectral editing, which obviously started with GABA, which you have to target by shooting an editing pulse at this frequency. Then there was glutathione, where you shoot an arrow essentially there. There's ascorbate or vitamin C. There's this NAAG that I was talking about. There's aspartate. There's lactate over here. And this tumor marker 2HG. And all of these would be exciting to look at. But again, you technically have to run a separate experiment for all of these. So a question is, how far can we take this multi-metabolite approach? Can we measure all of these in the 10-minute experiment reliably? And fortunately, we found a way how to do this, because we just add a bunch of editing pulses that all cover essentially the frequencies that we need in order to manipulate these signals. And again, I'm not going to go into too many details of the reconstruction, but what we finally end up with are essentially 3 different spectra, one that contains the major metabolites, one that contains GABA, glutamate, and so on, and one that contains, well, glutamate, ascorbate, aspartate, NAAG, and lactate over here. There's not a lot of overlap. We separate overlapping metabolites into different subspectra, and we quantify them with a linear combination modeling approach that we get from quantum mechanical density matrix simulations. So we know what the signals are supposed to look like in this experiment, and we just chase it through a fitting algorithm in order to tear them apart. So that's it from the acquisition side, and I think I have like 3 or 4 minutes left. So I want to spend a couple of minutes on that, on something where MRS has been lagging behind a couple of the other MRI disciplines, in my opinion, and in the opinion of a couple other people too. The problem is that right now everybody is essentially doing their MRS as they see fit. There's very little standardization in terms of acquisition. There's very little standardization in terms of data analysis. There's very little consensus on how to do this, and there's very little openly available code if you want to start analyzing some data, which is weird because MRS preceded the actual imaging part of the application of the NMR effect. Spectroscopy was the first thing that people did. But to be honest, we haven't made that much progress. And I firmly believe that this heterogeneity is kind of hindering the MRS breaking into mainstream. I remember that every time we go to ISMREM, they put us in the last dark corner of the exhibition hall, a couple of posters, and oh, there's the ugly stepchild in the back there. And all the other deep learning people, they get their massive 5,000 people keynotes, and yeah, well, we're getting a couple of posters. Huh?
It's all based on single noise.
It's not wrong.
GABA was 80 millimolar concentration.
Yeah, I can measure that easily. Yeah, correct. That's why we're here. That's why we're trying to get better. So one reason is specifically for spectral editing. By now, you can get an implementation from your vendor. If you have a Siemens scanner, you just ask your Siemens representative, and they have a WIP sequence. The problem with this WIP sequence is, and I hope I don't step on anyone's toes, but it's pretty terrible, because it turned out that in order to ideally measure GABA, you have to measure it at 68 milliseconds, and you have 2 editing pulses, and they need to be exactly half the echo time apart, which already isn't the case in the Siemens implementation. Now, if you want to do lactate, for example, you will choose a longer echo time of 140 milliseconds sometimes, then your editing pulses need to be 70 milliseconds apart. Guess what happens if you do that in the Siemens sequence? The editing pulses stay exactly 25 milliseconds apart. It's terrible. And you can see here, if you try and measure lactate with Siemens or Canon, your well, editing efficiency and your signal that you get out of this just goes down dramatically. What we've come up with as a solution was we have teamed up with a couple of people who work at Siemens and at Philips, and we have obtained a little bit of source code. We've come up with pulses of our own, and we have implemented one sequence that has the identical timing, the identical pulses across all 4 vendors. And now the signals look pretty much alike. It is more obvious for the lactate editing than it is for the GABA editing, which is already pretty okay. But still, we've managed to improve the agreement slightly. And this sequence we've already distributed to Eva, for example, so that is available on the scanner here, on one of these scanners. What is going on? All right. And finally, one tiny little thing that is very dearly to me, myself, is data analysis. So some of you have tried analyzing MRS data. You pointed up, how do you analyze your MRS data? Okay. Okay. That is one approach to do it. I'm sure if I asked 5 more people, I would get five more different responses, right? So, and I guess they inherited it from somebody who was there before. So it's always a little bit like it's wisdom from the ancient that's being handed down within the lab. It's like the holy grail that's being passed on. Generally, what we can always break MRS data analysis down is 3 steps that are essentially pre-processing, then quantifying, modeling and quantifying it. So if you look at pre-processing, maybe you are good at MATLAB and you write your own code, or you find somebody in your lab who knows what they're doing, or maybe doesn't know what they're doing, and they just hand down some code, and you use it, and it may or may not be right, it may or may not be good, who knows? Maybe you don't pre-process your data at all because you rely on the online scanner reconstruction on the clinical scanners. You may lose a little bit of data quality on that. But the bottom line is everybody is doing it differently, right? There's no standardization. People don't report it. People don't share their codes. Try looking for standard MRS processing pipelines online. You won't find any. And who's to say that out of all the 2,000 whatever MRS papers that have been published, all of them have used the perfect processing code? I'm not willing to buy that. Specifically for human data, what people do, and also for animal data, you always have to prepare it into a weird format, export it into a software that runs on either on a server or somewhere else. It does some oblique things that nobody really understands, because LT model is a commercial product, for example. One license costs $17,000, and the guy who runs it has published it 27 years ago and hasn't made many changes ever since. So again, it's all very opaque. You can tweak a little with the settings. You can almost get any result that you want. We don't really know what these things are doing, which is why we don't really understand where they're going. This is not really how quantitative data analysis nowadays should be done. Finally, you feed it back. You may want to do some tissue correction to allow for the fact that there's gray matter and white matter in your voxels, obviously. And for that, some people have their own code. Some people have an Excel sheet. That is actually true. There is an Excel sheet floating around on the internet that Paul Mullins has created once, and it's widely being used. Who knows what relaxation times Paul is using for that? It's just, again, it's tedious, it's error prone, and it's overall just not the way that things should be going. So we've come up with a successor to the Gannett software that was specifically designed to do GABA spectroscopy, and we're staying in theme because my supervisor is a bird enthusiast. We've called it Osprey. And Osprey essentially takes your data and shoots them through these 7 parts of the pipeline, where you don't have to do anything much else. And the first step is pretty much to define what you're going to do. So there's a job system. It's very, very similar to what SPM is doing. So I don't know whether some of you are doing fMRI analysis with SPM. You create a job file where you tell the program what you want to do. And you do the same thing for Osprey. You tell them, well, find the data here, find some water reference data here, find your structural images there, set some options, and go. So it creates a data container in MATLAB where it saves all the data. If you want, you can also feed that job as a CSV file if CSV is your thing. That's fair enough. Then there's the load module that just acts on the container that you just created. So it automatically pulls in all the data, it recognizes the file format that you've provided. It does the coil combination for you if you happen to have multi-receiver coil data. It's all happening automatically without you having to do anything. Next module is the process module. It does all the steps that are necessary to create the final spectrum that we're going to feed into our fitting algorithm. So there's individual alignments of the individual averages that you get. Do an eddy current correction. We do frequency referencing. I apologize for the resolution of these graphics. You get a final spectrum that is, well, under any given circumstance, the optimal quality that you can get out of this data. If you do mega editing, for example, you get a different spectrum. You get the sum spectrum that we can feed into the analysis. There's a fitting algorithm where we fit the real and the imaginary part of the spectrum at the same time. And we do that based on a basis set that we've simulated. We allow for first order phase correction and a zero order phase correction. We allow for some line broadening, a little bit of frequency wiggle, then obviously the amplitudes are model parameters. And we have a baseline that is very similar to what LC model does. It's a smooth spline baseline. And essentially this is all a non-linear least squares algorithm in order for us to figure out how each of these basis functions contributes to the final spectrum. This is very similar to what LC model does. We're trying to reverse engineer it. We get some pretty nice fits here, as you can see. The visualization is so that you can see which of the metabolites contributes how much to the final signal. You can do the same for GABA-edited spectroscopy. And we're trying to, or well, we're currently implementing it for all the other sequences that we have developed so far. There is a, currently, well, there's a couple approaches to model that. And the idea behind the software is that it makes it easier to try all of these, to compare and actually find out what is going on and how do we actually accurately do it the best way. It is supposed to be crowdsourced and open source so that people can modify their things other than LC model, which is very much tied to whatever this person who's sitting in Ontario in his lake house has produced. To answer your question, you can either let the baseline figure it out, or you can add specific macromolecule basis functions to it. There's several ways, and the good thing about this is that you can try new ones. Yes.
Can we use our own basis sets?
I am in the process of writing a tool that allows you to import an LC model basis set, for example. Other than that, if you have simulations with FIT-A, for example, you can. There's an importer. Yeah, that is the case.
They change a lot as a bunch of fields.
Excuse me?
This is all 3T.
This is at 3T, but the tool can handle any field strength as long.
When you do simulations.
Yeah, correct. I mean, yes. So you can easily simulate something at 7 or 1.52 and just import the basis set. Yeah, that works. It's just like LC model in that case, where we're not tied to a field strength. As long as you have the simulations, which can be done in with the same toolbox that this is essentially relying on. It also can read out, well, your voxel position and everything to co-register your voxel and create a tissue composition mask. And it derives a number of quantitative parameters, creatine ratios, raw water scale, then CSF corrected, and fully tissue and relaxation corrected. You can enter your own relaxation values if you want, but there's a library built in. We have recently come up with a GUI where you can do some data quality assessment, pre-alignment and post-alignment. You get an impression of the drift that has occurred during the experiment. You see what your final spectrum looks like. And again, you can assess the quality of your fit and you get the quantitative results right away. And the most important thing, I think, in the design of this is that it's all supposed to be out in the open for everybody to modify. So we've put it all on GitHub. There's no hidden algorithm anywhere. There's no external libraries. It's all running in MATLAB. And we want people to be able to look at what we're doing and to be able to tell us, look, this is dumb. You should do it differently. This is part of how we make these things better, is not by relying on somebody who's developed an algorithm 27 years ago. All right, let's come to an end. I've talked for a little bit too long already. So generally, I hope that all of you remember from this talk, spectral editing opens up the observation of a bunch of molecules that would otherwise not be really accessible to us. We can measure them, we can quantify them. Specifically, GABA levels. we can measure. They are functionally, behaviorally, and clinically relevant. And we have developed a couple of new acquisition techniques that allow us to do these things more efficiently, more rapidly, gain more information from a single clinical scan. Hopefully we can do all that in a more standardized and more comparable way across scanner ecosystems and across sites, because we need more data, obviously, in order to answer the important questions in research. And if everybody publishes studies of 10 subjects that they've run on their single scanner, we're not going to be able to answer these questions, I believe. Generally, it's a cool tool. If you know how to use it, can bridge a scaling gap somewhat, helps you answer questions that are not easily answerable with normal imaging. We can greatly expand its functionality, but we need, as a field, to move this thing towards more collaboration, to being more open, and to further standardize what we're doing, because otherwise I think this is not going to go very far. And with that, I'd like to acknowledge funders and collaborators and our wonderful bird logos of our previous software versions. We're staying in the theme, and you can look it up. The GitHub repository is behind this tiny URL link. I'm posting updates on Twitter every now and then, and if you have questions, you can always send an email as well. And with that, thank you for your attention and your frequent questions. Thanks.